Welcome to the Sand Science Experience. Today we're diving deep into some really fresh research. It's a brand new study, super comprehensive, looking at our cells' powerhouses, mitochondria.
That's right.
And specifically, how they behave across a whole bunch of different tissues in mice.
Yeah, and what makes this one stand out, I think, is that it's not just about the mitochondria themselves.
Right.
It digs into how things like sex and, importantly, age affect how they work.
Exactly. So it's like getting a map of how our organs might change as we get older, which, is so critical because aging is linked to so many diseases.
Absolutely. Focusing on mitochondria gets us right down to the energy level of how things change. Are there different patterns for males and females as they age, that kind of thing?
Okay, so the big question we're tackling, based on this amazing study, is how does mitochondrial function actually differ between, say, your heart and your skin? And how do sex and age tweak those functions?
And this study is generating what they're calling the most extensive data set we have on this. It's a really deep look.
An unprecedented view, really. So if you're curious about how aging works at that fundamental cell level, but maybe don't want to wade through dense papers,
you're in the right place. We're going to pull out the key stuff, the surprising bits.
Yeah, definitely. And hey, if you like what we do, please remember to like and subscribe. It genuinely helps us keep making these deep dives for you.
It really does.
Okay, so let's get into the nuts and bolts. What did these scientists actually do? It sounds massive.
Oh, it was. It was a Herculean effort, like you said.
Yeah.
They measured respiratory activity. That's basically how mitochondria produce energy.
Okay.
And they did this for mitochondria from 33 different tissues in mice.
43, wow.
Yeah. And they looked at two key points in life. Young adults, about 2.5 months old, and then old age, around 20 months.
And both males and females in those groups?
Both males and females, yep. Crucial comparison.
So 33 tissues, 2 ages, 2 sexes. That's a lot of samples.
It was staggering. 1,320 tissue samples in total from 40 mice. And get this, for reliability, they tested each sample 3 times.
3 times?
3 times. So you're talking nearly 4,000 individual lab assays.
Okay, that's commitment. How did they even manage to compare that many tissues fairly? Was there some new technique involved?
Yes, and that's a key part. They used this innovative method that lets them assess mitochondrial function in tissues that were actually frozen beforehand.
Ah, okay. So they didn't have to rush to analyze fresh tissue.
Exactly. It's a big deal because it means they could collect everything, freeze it really carefully, and then run the analyses together later. It helps minimize, variations you might get running things on different days.
That makes a lot of sense for consistency. So these respiration assays, What are they actually measuring? Oxygen?
Pretty much, yeah. They measure oxygen consumption, which tells you directly how actively the mitochondria are generating energy.
Right.
And they were clever about it, too. They used specific chemicals to stimulate different parts of the energy production line complexes 4 and 4V to see how each part was functioning.
Okay, got it. Stimulating different parts of the engine. So this mountain of data, what were the big takeaways? Anything surprising jump out?
Well, one of the first really striking things was just how much mitochondrial activity varies between tissues, even in mice of the same age and sex.
So your heart mitochondria are just built different from your, say, skin mitochondria?
Fundamentally, yes. Different tissues, different energy needs, different mitochondrial capabilities. It really hammered that home.
Can you give some examples? Which tissues were the powerhouses?
Sure. Tissues you'd expect to work hard, like the heart, obviously. Also brown fat, that's the heat-generating kind, in the kidney. They consistently showed the highest respiration levels.
Okay, it makes sense. High-energy jobs. What about the tissues on the other end? Lower activity.
On the lower end, they saw things like the colon, the plantaris muscle, that's a specific one in the leg parts of the small intestine, like the jejunum and ileum, and also mesenteric white fat, consistently lower activity there.
Interesting. And you mentioned the plantaris muscle. So even within muscle types, there are differences.
Exactly. That was another point. Even within, say, different brain regions or comparing various skeletal muscles, you could see distinct mitochondrial respiration signatures. It's really fine-tuned.
Okay, let's switch gears a bit. What about sex differences? Did they find significant differences between young males and females?
They did find statistically significant differences related to sex, yeah. But overall, when you looked across all tissues in the young mice, sex was, let's say, less dominant factor than just which tissue you were looking at.
Oh, okay.
And the key thing is these sex differences were very tissue specific. not like a blanket male mitochondria work this way, female mitochondria work that way.
Right, more nuanced. So where did they see the biggest differences in young mice?
Okay, let's break it down by complex. For complex 1, respiration was higher in young males in the quadriceps muscle and gonadal fat. For complex 2, males were higher in kidney medulla, stomach, quad, hippocampus in the brain, and another leg muscle, the gastrocnemius, but lower in gonadal fat and the ilium.
Quite a mix.
It is. And for complex 4, again, males higher in brown fat, heart ventricles, kidney medulla, hippocampus, stomach, quad, gastrocnemius, but lower in gonadal fat and the duodenum, part of the small intestine.
Wow, that's specific. Was there any kind of general pattern, though? Like overall trends?
Yeah, sort of a general trend emerged. Young males tended towards higher respiration in stomach, kidney, and skeletal muscle. Young females, on the other hand, tended to be higher in parts of the GI tract and certain types of fat.
Suggesting some early specialization, maybe?
Possibly, yeah. Early divergence in energy management strategies in specific tissues based on sex.
Fascinating. Okay, now the big one. Age. How much did things change between the young and old mice?
Age. This study really drove home that age is the dominant factor. It had a much bigger, more widespread impact on mitochondrial activity across most tissues than sex did.
Really? More than sex?
Much more, yeah. The footprint of aging on these cellular power plants seemed significantly larger.
Which tissues showed the biggest age-related changes? Where was aging hitting hardest, mitochondrially speaking?
They saw really substantial changes in most brain regions they looked at, various fat depots around the body, different skeletal muscle groups, the eyes, and also different parts of the GI tract. It's quite a broad list.
So aging isn't uniform across the board, then?
Not at all. Different tissues seem to age at different rates, at least in terms of their mitochondrial energy production.
And did these age effects play out the same way in males and females? Or were there differences there, too?
There were differences there, too. The study highlighted that the effects of age could be, well, both sex-specific and tissue-specific.
Okay, so it gets complicated.
It does. The way a tissue's mitochondria aged could depend on whether the mouse was male or female. Some of the biggest overall age effects were seen in places like the pancreas, heart, fat tissue, and skeletal muscle.
Interesting. Some overlap with where the sex differences were seen earlier. What about tissue resilience? Did anything seem to resist aging effects?
Yes. That was a really cool finding, actually. While lots of tissue showed changes, they did find some tissues that were remarkably resilient. Their mitochondrial activity stayed relatively stable even in the older mice.
That's hopeful. Makes you wonder what they're doing right.
Exactly. What are the protective mechanisms there? That's a huge question now.
Okay, let's dig into those sex-specific aging effects. You mentioned divergent responses.
Right. So looking overall, both males and females tended to show a relative increase in complex activity with age and a decrease in complex 4 activity.
Okay, some shared trends.
But complex 2 showed this really interesting divergence. Its activity tended to go up with age in males, but down in females.
Wow. Completely opposite directions for a key part of the energy pathway.
Exactly. Suggests males and females adapt or, well, deteriorate differently in that specific process as they age.
Where were these sex-specific age effects most pronounced? Which tissues showed the biggest divergence?
For complex 1, the biggest sex differences in aging were in the stomach, jejunum, distal colon, quad muscle, and pancreas. All showed divergent changes. For complex 2, it was stomach, duodenum, distal colon, pancreas, jejunum again. And for complex 4, mesenteric fat, duodenum, jejunum, skin, and distal colon showed the biggest sex-divergent aging effects.
Lots of action in the gut and pancreas there. Were there any tissues where aging looked similar for both sexes for specific complexes?
Yes, there were. For complex I, for example, gonadal white fat, the kidney cortex, that's the outer part, and the diaphragm muscle, showed pretty similar age-related changes in both males and females.
So in those cases, age seemed to be the stronger influence, overriding sex differences for that specific function.
Seems that way, yeah, for that particular step in energy production in those tissues.
Okay, so it's this complex tapestry of shared and distinct aging patterns. What about the overall direction? Was it mostly decline, or were there increases too?
Both, actually. That was another surprise. Some tissues showed a shared increase in respiration across all 3 complexes with age.
Increase?
With age?
Yeah. Like the plantaris, diaphragm, and soleus muscles, all skeletal muscles. Also the heart ventricle, parts of the digestive tract like the ilium, cecum, proximal colon, and mesenteric fat. all showed this consistent increase.
That is counterintuitive, especially for muscle.
It really is, especially because other skeletal muscles, like the gastrocnemius, hamstring, and tongue, show the opposite, a reduction in respiration with age.
So even within skeletal muscle, it's not one simple story.
Not at all. Very diverse effects of aging.
Okay, what about tissue showing that more expected pattern, a consistent decrease across the board?
They saw that shared decrease across all three complexes in brown fat, gonadal fat, inguinal fat, so various fat types. Also, most brain regions they looked at: cortex, hippocampus, hypothalamus, cerebellum.
Most of the brain declining, okay.
Right. And also liver, kidney cortex, note, different from the medulla we mentioned earlier, the eye, skin, and spleen. So lots of key areas showing that general decline.
Highlighting again how tissue-specific this all is, even within one organ like the kidney.
Precisely. So putting it all together, what's the headline? The main conclusion from this massive study.
The big headline is definitely that age has a much larger, more pervasive effect on mitochondrial function across tissues than biological sex does.
Okay.
And they really emphasize the strength of this conclusion because of how rigorously they did the study: uniform collection, standardized analysis, huge sample size, high confidence in the data.
And does this fit with other aging research, like studies looking at genes?
It does, yeah. It aligns well with transcriptomic studies looking at gene activity, which also show that different tissues kind of follow their own aging paths genetically. This mitochondrial data adds a crucial functional layer to that picture.
So what are the big picture implications? Having this detailed map, what does it help us do?
Well, it's an incredible resource for understanding how age-related decline might happen in specific organs. If you know which tissues and which mitochondrial processes are most vulnerable,
You can start asking why and maybe how to intervene.
Exactly. It helps guide future research into the underlying mechanisms, and you can start thinking about potential therapies targeted to specific tissues or mitochondrial pathways to promote healthier aging.
Did they speculate on why these changes happen? What might be going wrong in the mitochondria?
They touched on likely suspects, things like lower activity of the genes that build the energy machinery, the OXPHOS system, and also the known accumulation of damage to mitochondria over time.
Right, the wear and tear.
Yeah. But they were careful to say, more research is needed to nail down cause and effect. It's complex.
And what about those weird increases in respiration and heart and some muscles with age? That still seems odd.
It does. Their speculation was maybe it's a compensatory thing, like if the tissue mass or overall efficiency is declining, maybe the remaining mitochondria have to work overtime to keep up energy production.
Like they're revving higher to compensate for fewer workers or less efficient ones.
Sort of, yeah. But again, that's a hypothesis that needs more investigation. What signals would drive that? We don't know yet.
Well, this has been a fantastic deep dive, really detailed stuff. So, just to summarize the key takeaways for everyone listening.
Okay, main points. 1, mitochondrial energy production is highly tissue specific. Your liver is not your brain, mitochondrially speaking.
Got it.
2, age is the biggest factor driving changes in mitochondrial function across the board, more so than sex.
Age rules.
Pretty much. And 3, how aging affects mitochondria isn't uniform. It can differ between tissues and even between males and females for certain tissues and certain mitochondrial processes. It's intricate.
It really paints a picture of the incredible complexity of aging, right down at the cellular engine level. This atlas sounds like a goldmine for researchers.
Absolutely. A foundational data set for understanding aging mechanisms and thinking about future ways to help people age healthier.
So a final thought for you listening. Think about how these microscopic changes in mouse cells might relate to the bigger picture of aging we see. Based on this, which tissues might worry you most about energy decline as we age? And what new questions does this spark for you? Thanks for joining us on the Sand Science Experience.
Thanks, everyone.
And please, if you found this interesting, do hit that like button and subscribe. It really helps us out. See you next time.